So our second speaker in this panel is going to be Jochen Ditterich. He's an associate professor here at UC Davis in neurobiology, physiology, and behavior, and is a member of the core faculty at the Center for Neuroscience. His research focuses on neural mechanisms underlying higher-order brain functioning that link perceptions to action, and specifically focusing on decision-making. And so today he's going to be talking to us about computational mechanisms of perceptual decision-making and their neural implications. Thank you, Jochen.
Yeah, well, thank you very much, and good morning, everybody. So our decisions are influenced by a large number of factors, right? Sensory information about the current state of the world, expected outcomes or costs of potential options that we could choose, past experience, emotional state, and so on. And so some of these are really hard to measure and quantify. So if we want to understand how our brain makes decisions, we actually need to study well-controlled decision tasks. And what we actually focus on currently, therefore, is perceptual decision-making. So these are decisions that should be purely informed by sensory information, which we have very tight control over when we are running an experiment. So in addition, if we also want to understand how the computations that are underlying decision-making are actually implemented in the brain, we need to be able to record neural activity during such decisions with very high temporal and spatial resolution. So why is that? Well, most straightforward decisions that we are making are made within a second, perhaps even less. So if you want to see neural activity unfold, you actually need millisecond resolution for those measurements. Likewise, any kind of technique that would only tell us what the averaged activity of a larger pool of neurons in some area in the brain is would not allow us to track individual neural signals that are underlying decisions. So since none of the currently available non-invasive techniques allow us to make measurements with such precision, what you need to do is you have to use invasive recording techniques, and therefore you have to do animal studies. Okay, so if you want to do animal studies, you obviously need a decision task that can be learned by animals, right? And in the end, what we would like to have is a task where we have comparable behavior between humans and animals, because we want to use our animals as a model for human brain function. Also, the task should involve well-characterized input and output brain circuits, so those that are well studied, and then we can actually focus on the decision process. And based on all of these reasons here, we've chosen a particular experimental framework, which is visual random dot motion discrimination with goal-directed eye movements for reporting the choices to study decision-making in both human and non-human primates. And the reason here is we actually understand quite well how our brain processes and represents visual motion information, and we also know quite well how eye movements are controlled and processed. Okay, so let me give you an idea of what that basic task looks like. So we have a subject looking at a computer screen, and on that computer screen we are presenting a so-called random dot motion stimulus. So that is a certain number of little flickering dots that are shown within a circular aperture. And what we can do is we can embed some motion signal into that flickering by having a certain fraction of those dots move coherently together in a particular direction. And so in the simplest case, you could imagine that there are only two possible directions of motion. Let's say there's either some motion to the left or some motion to the right. How many of dots are moving together is something that we can control as the experimenter, and that makes the decision either easier or harder. And in our experiments, that changes randomly from trial to trial. Okay, so now the observer would look at that stimulus, and they can look at it as long as they want to, and they try to make up their mind whether there is some motion going to the left or to the right. So once they think they know what's going on, they make a goal-directed eye movement to one of two possible choice targets shown in red here. So if you believe that there is some motion to the left, you would actually look over at that left target. If you believe that there is some motion to the right, you would actually look at that right target here, and we can measure choices and response times. Okay, let me show you an example of what such a stimulus looks like, and so you can try to figure out, let's see. Let's go back. Let's see whether I can get the cursor here. So is that the right screen? How can I actually move the cursor on that one? Oh, there it comes. Okay, thank you. All right. Okay, so you can watch the stimulus now and try to figure out what that direction of motion is. Or not? Okay. Good. So any guesses? Right. Okay, so that was a relatively easy one. So in this case, actually half of the dots were moving together in a particular direction, right? So this is what it looks like when we make your job a little bit harder. In this case, only 10% of the dots are moving together. So any idea what was going on here? It actually was right again, right? But you can see that this was a much harder decision than the first one. Okay, so what does the decision behavior in such a task look like? I'm going to show you the monkey behavior, but if you do it in humans, it virtually looks identical. So the first one we're looking at here is the accuracy of the decisions as a function of how much useful information we are providing in the motion stimulus. And so if the motion is strong, the monkeys pretty much always get their choice right. As you provide less and less useful information, accuracy declines, and you have to end up at choice performance when there is no useful information available. So what about mean response times? Turns out that that also systematically depends on the stimulus. So if the decision is easy over here, if you provide a lot of useful information, choices are on average pretty fast. Here we're close to something like 400 milliseconds. If you present less and less information, you can see that the monkeys are slowing down more and more and actually take more time to make up their mind. All right. Good. So now, as I told you, right, while these decisions are made, we actually record activity from inside the brain. And what I'm going to show you is that you can find a neural correlate of the decision process in parietal association cortex. So the particular area that we are recording from in this case here is the lateral intraparietal area in the intraparietal sulcus, also called area LIP. And what we know about neurons in that area from previous work is that these neurons are implicated in visuospatial attention and the planning and execution of saccadic eye movements. We know that those neurons have spatial response fields, so that means that they are only interested in particular regions of visual space. And a certain subpopulation of those neurons has an interesting property. They actually maintain continuous activation during a so-called memory-guided saccade task. So what you do there is you just briefly flash a visual stimulus and you ask the monkey to remember the location of that stimulus in order to make an eye movement to that location later. And what you find then is that these types of neurons actually hold on to the location information of that stimulus during the delay period and allow the monkey to later execute that eye movement. And these are the neurons that we are recording from here and that I'm going to tell you about. Okay, so the task then is set up in such a way that we place one of the two choice targets inside the response field of such an LIP neuron. And a monkey obviously can either choose that target inside and make a saccade there, or he could end up choosing the other target and make a saccade to the other side. Good. So let me now actually demonstrate to you or play you an example of what those neurons do. Okay, so in this case, you're going to see what the monkey sees on the screen, the motion stimulus and the choice targets. You can also follow where the monkey is looking at on the screen currently, and that will be plotted in blue. So you see basically when the monkey makes his eye movement, and you will be able to listen to the activity of one of those LIP neurons. In this case, actually, when the monkey will end up choosing the target inside the response field. Okay, so let's see if I can get my cursor back. Yeah. Nope. Still playing. Okay, so what you've hopefully heard, right, was that that neuron was increasing its activity over time, right? And what you might have noticed too is that that increase actually happened quite a bit before the monkey actually made his eye movement, right? Okay, let me show you a second example. So now in this case, the monkey will actually end up choosing the other target outside the response field of that neuron. Let's see, go. Right, So eye movement was made now. You should have heard that that neuron was actually decreasing its activity in that case, right? And again, that change was happening quite a bit before the monkey actually made his response, so, which is kind of cool, right? By listening to those neurons, you can make a prediction of what the monkey is going to do sometime later, right? Okay, so now if we record from a larger number of those neurons and we average their activity over many decision trials, what do we see? Well, it actually turns out that early on during a decision, the activity of those neurons is largely determined by the visual stimulus that we are showing to the monkey. Okay, so in this case, you can see that for approximately 200 milliseconds, there's a very stereotyped response in those neurons. And after that, we see a ramping response where we have either increasing firing rates, shown here in these solid traces, or decreasing firing rates, shown with the dashed traces. The solid traces are for visual stimuli that had motion in a direction that would support choosing the target inside the response field, and the dashed traces are for visual stimuli that had motion in the opposite direction, so where it would be better to choose the target outside the response field. What you can also see is that the slope of those ramps here systematically depends on the strength of the visual motion signal. So red is a very strong signal, there we have the steep slopes, and here you can look at weaker signals. If you look at the activity of those same neurons later on towards the end of a decision, and if we sort those trials by the choice that the monkey ends up making, this is what we see. So now all of the solid traces are for trials where the monkey ended up choosing the target inside the response field, and all of those dashed traces here are for trials where the target outside was chosen. What you can see is that in the case where the target inside the response field is chosen, the activity reaches a very stereotyped activity level just prior to that choice. And also, the strength of the visual stimulus that was informing that choice is pretty much lost in the neural activity. In contrast, when you're looking at the responses down here, right, choosing the target outside the response field, there's still a remaining representation of the strength of the sensory stimulus that led to that particular decision. Nonetheless, the main determinant of the neural activity towards the end of the decision is the binary choice that the monkey is making. All right, so to summarize so far, I've shown you that the LIP neurons show a gradual shift from representing sensory evidence to representing categorical choice. I've also shown you that they have a ramping activity in response to the visual stimulus, right, with firing rate increases or decreases according to direction and strength of the visual motion. And I've shown you that there's a very stereotyped firing rate immediately prior to choosing the target inside the response field. Okay. So the question now is, can we actually explain potentially how those decisions are made? And what I'm going to show you is that we can actually have a computational explanation for what is probably going on here. So what you're seeing here is the structure of the computational model that will be able to explain the decisions. So we are looking at decisions between two alternatives in this case, right? And so either picking the right target or the left target, right? What you see shown in green here are two integrators. Each of them is associated with the two possible choices, and they actually accumulate evidence for whether it would be a good idea to make a particular choice. Okay. So the output of those integrators is compared to some decision threshold. So the idea there is, you can think of this as a race between two representations of the two possible actions that you could take, and whichever of them reaches a critical activity level first would terminate the decision and determine the choice that is made in that particular case. And so the question now is, what is actually integrated by those integrators? And in our case, we're making decisions between directions of motion, either left or right. So in this case, we start out with pools of sensory neurons that are either tuned to rightward or leftward motion. And the proposal here is that the difference between the activity of those two pools has to be calculated, and that difference is then feeding into those integrators. Okay, so can a model with that structure explain the decision behavior? What you're seeing here is, again, the decision behavior that I had shown you before, shown with the symbols, the black symbols. And the lines are actually the predictions of the best fitting model with the structure that I had just shown you before for what that decision behavior should look like. And you can see that it can capture the accuracy of the choices very well. It can capture the mean response times. And in addition to that, there is a phenomenon that I haven't told you about before. And this is on average, when our monkeys are making errors, they're a little bit slower compared to when they make correct choices. And this is something that our model can actually explain as well. Okay. So in addition to that, also when you're making decisions, your decision time varies somewhat from trial to trial. Sometimes you're a little bit faster, sometimes you're a little bit slower, and that leads to certain distributions of the response times shown here with the gray histograms. And the colored lines that you can see are again the predictions of the model, what those distributions should look like. And you can again see that those are captured very well. Okay. So now the model can explain the behavior, but can it potentially also explain the neural activity? So in order to get at that, what we're going to do is look at the predicted output of those evidence integrators here and compare them to the neural recordings that we have done in parietal cortex. So important to keep in mind, the model is only based or fitted based on the decision behavior, right? So we are not using the neural data for that. We're just using a prediction of that behavioral model to compare it to the neural recordings. And when we do that, you can actually see that there's a relatively good agreement again. So what you see in the little dots here are the actual neural activity that I've shown you before that has been recorded, and the lines are the predictions of what that should look like according to the model. So this is for the early activity. If we look at the late activity, right, again, we have a pretty good match, and the major features of that activity are captured. Okay. So far, so good. So now you might ask, well, why should the brain integrate the difference between the activities of two pools of sensory neurons until some decision threshold is reached, right? Does that make any sense? Okay. And so therefore, we have to look at the relationship with the mathematical theory of optimal decision making. So going back to base, when you want to make a decision between two different alternatives, here hypothesis H1 and H2, based on a certain observation, what you should do is you should look at the conditional probabilities of making those observations in those two different states. This is what we call the likelihood ratio. And you should also take into account the prior probabilities of the two possible hypotheses. In our case, they are the same, so for our purposes, we can forget about those for now. If you make multiple observations over time, and if those are statistically independent, you should actually multiply all the likelihood ratios of those observations with each other, and then in the end, you should make a decision based on whether that ratio is larger or smaller than 1. There's one more thing that you can prove, and that is actually if you want to minimize your average decision time for any given desired level of accuracy, what you should do is collect information until that ratio here reaches a particular constant, a particular criterion. Okay, and that is what is called a sequential probability ratio test. Good. So now integrating or basically collecting evidence until a certain threshold is reached is something that we like, because I showed you in the model, right, that we have a decision threshold, and once that is crossed, we terminate the decision and trigger a particular choice. What we don't like so much is the multiplication here. There was no multiplication in our model, right? Okay, so equivalently, you can look at the log likelihood ratio, okay? And if you do that, you actually want to sum those across the different observations, right? And summing something over time is temporal integration, okay? So we like that a little bit better because we had temporal integrators in our model. So the last thing that is missing, that would only make sense if what we are integrating are log likelihood ratios, okay? And so without being able to go into the details, what you can actually show is that if you have two samples, xi and yi, that are taken from two populations of sensory neurons that are tuned to leftward and rightward motion with certain properties that make sense biologically, you can actually show that the log likelihood ratio is proportional to the difference between the activities in those two pools. Okay? So what that shows overall is that the brain has actually found a very clever approximation to the statistically optimal algorithm that you should use for making your decisions in that case. All right. So I'll skip that summary because we are getting short on time, and I'll just briefly touch upon a couple of the more recent studies that have used more advanced versions of that framework. So we've addressed then how the decision mechanism can actually generalize to situations where you're making decisions between multiple alternatives, not just two. That's interesting also from a theoretical point of view, because it turns out that basically the equivalent of the SPRT is actually no longer known for multi-alternative hypotheses. So we really don't know what the general best possible algorithm is to use when you're making decisions between multiple alternatives. Also, what I've shown you so far was only that the average neural activity, right, over many, many decision trials looks like we are accumulating evidence over time, right? So, but what is really happening during individual decisions? In order to get at that, it turns out that just recording from a single neuron is not enough. You don't get enough information from that. So you actually have to start recording simultaneously from multiple decision-related neurons, which we've done. You can show then actually that the population responses are still consistent with this integration to threshold idea, even on individual decisions. And you can even show some more interesting things. So for example, you can identify changes of mind based on the neural activity, something that you would never see in just a decision behavior. And here's just one brief example of that. So you're looking at a simultaneous recording from 4 decision-related neurons in the same area that I had talked about before. The ones in the first 2 rows are associated with one potential choice. The ones in the 2 bottom rows are associated with another choice. All those vertical lines are individual action potentials that are fired by those neurons. And here in the end, you can see that those 2 neurons were the most active, right? The other ones were silent. And this is actually the choice that the monkey ended up doing at the end of the trial. Nonetheless, what you can see is that earlier on, the other 2 neurons were very active. And in fact, the state decoding shown in blue here actually shows you that they basically were as active as they should normally be just before that particular type of choice here, right? The other neurons were actually inactive. And so we believe that temporarily the monkey was actually favoring a different option here and later changed his mind. So we don't have time to go into that, but we did a couple of controls actually to convince us that we are not just looking at random fluctuations of the neural activity here. Okay. And so just last thing, what we're doing right now is we're looking at decisions with more flexibility. So we are addressing questions like, how can you actually separate decision relevant from irrelevant information when you're making those decisions? And also, how can the brain switch stimulus-response associations when the context of the decision requires you to do so? Okay, and that's it for now. So if you're interested in more detail in any of those, please feel free to ask during the discussion part of the panel. But for now, thank you very much for your attention.